We are from group 19 and throughout this video, we're going to detailing the journal entitled Genome Sequence of the Model Rice variety Kita-Aki X. And here is our group member.
As we all know, rice provides foods for us human and also the other living things, and it is used as a model organism for functional genomic studies. There are more than 3,000 rice varieties and species were found by researchers. However, there are the downside in functional genomic studies, which those rice variety have long life cycle and also low transformation efficiency. So, the Kita-K cultivar has emerged as the greatest model for rice research because of its properties, such as insensitive to daylight, easy to propagate, relatively cold tolerance, short in statures, and also complete its life cycle in about 9 weeks, which is shorter than the other rice varieties. Kita-K cultivar is being used to explore diverse aspects of rice biology. Kita-K cultivar is being used to explore diverse aspects of rice biology, including flowering time, disease resistance, small RNA biology, and also CRISPR-Cas9 and talent technologies. Kita-K-X is a Kita-K plant that carrying rice AX21 genes. The establishment of the high quality genome of Kita-K-X used to compare to other rice variety to identify the genomic
I will
variation.
be presenting the methodology. The plant materials used for the article is Kita-Ake, Zensha 97, Minghui 63, IR 64, 9311 and Kessler seeds. The construction of the phylogenetic tree is based on the genotype of all rice accessions from the SNP database RiceVar map version 2.0, where the IBS distance matrix was calculated and the construction of phylogenetic tree was by unweighted neighbor joining method. The high molecular weight DNA from Kita-K-X was isolated and used in sequencing for the genome sequence and assembly. The gene function was annotated from SwissProt and Tremble database using BLASTP, and the gene ontology term assignments, protein domains, motifs were extracted with Interpro, and the pathway analysis was derived from best match eukaryotic protein in KEGG using Circos. The genome synteny. The program used for genome synteny was SynMap to identify the collinearity blocks using homologous CDS pairs, and Circos to visualize the collinearity blocks. The repeat annotation was used with repeat masker and blaster to merge the output and to obtain fraction of transposable elements and repeated sequence. These were run using nucleotide libraries from RITEDB, and the custom proscripts was used for reconciliation of mass repeats. They identified the non-coding RNAs using RFAM library, and the transfer RNAs was predicted using tRNA scan SE. The analysis of genetic variations for SNPs and indels was used using MUMMR, and the analysis of PEVs and inversions was used with MUMMR2, and the inverted alignment blocks was used to identify inversions. The BAC library construction was used with established protocol, and the genome size estimation for the estimation of Kita-K-X genome size was used using Illumina fragment library with the histogram of 24 mer frequency, where the K-mer histogram indicates the peak value of particular
X
frequency.
flowers significantly earlier than the other sequenced rice variety, and under long day condition, we can see that Kita-K-X is heading at 54 days after germination, and the figure C shows that the Kita-K-X is related to other rice varieties using a phylogenetic approach, and we can see that there is 9 subpopulation that could be connected to its geographical origin. Kita-K-X and Nippon bear are the most closely related within the same subpopulation. Secondly, for genome sequencing and assembly, we can say that the high quality has been obtained by the de novo genome, and they used a strategy of combined short read and long read sequencing, and there the completeness is assessed by the alignment of annotated genes from B7.0 Nippon bed to Kitake X, and that shows a 98.94 completely aligned Kitake X assembly. And the bacterial artificial chromosome library was constructed and was sequenced, and the BAC clone shows in 0.09 overall error rate, indicating a high quality of this assembly. And for genome annotation, the predicted protein coding genes are distributed evenly across each chromosome. Gene density tends to be higher towards the chromosome end. And to evaluate the quality of annotation, they studied the conservation of functional genes in Kitake X. There's a genes from 3 different pathways, and it shows a 90% identity to the corresponding Nippon bed genes at protein level. So the results actually indicating a high quality of annotation. And lastly, the genomic variation between Kitake X and other rice varieties. So there is a genomic variation between Kitake X and Nippon bed that are highly concentrated in some regions, which can be seen in the and the variation between Kitake X and Zenshin are spread evenly throughout the genome can be seen found in C here. And for the variation occurring in the genome, they found that single base and 3 base pair without frame shifts are the most abundant than the others. That suggests genetic variation that has been functionally
Kitake
selected.
X, which is an early flowering rice variety with rapid life cycle that is easy to propagate under greenhouse condition. Protein coding gene of Kitake X contain 35,594. The availability of high quality genome and annotation for Kitake X will be useful for identifying genes controlling those traits. 219 SNPs and indels were identified between Kitake X and Kitake genomes, which resulted from somatic mutation that arose during tissue culture and regeneration, or maybe during spontaneous mutation. The mutation were identified by aligning the rates of Kitake X mutant to the Nippon Bear reference genome. 97 on the average, 97% of the Nippon Bear genome were covered by the Kitake X rates. In some regions, Kitake X genome diverged from Nippon Bear. This appears as a escape in coverage or as region containing a concentration of natural variation between Kitake X genome and Nippon Bear genome. Apart from the 97% of the coverage, we can use the Kitake X sequence as direct reference genome and detect the mutation in highly variable region. This will simplify analysis and increase the confidence in identification of FN-induced mutation. Lastly, Kitake X genome also can be used as a reference genome for Kitake.
Kitake X full sequencing genome project contribute to research field as it is served as useful reference genome for rice. Besides, it also accelerates functional genomic studies for rice and other varieties, as Kitake X can be used as reference. Last but not least, it also facilitates investigation into the genetic basis about diverse traits, which is very critical for rice biology and genetic improvement. Unavailability of full sequence genome of Kitake X can be posed as obstacle in rice research that use Kitake X in it. Such as example, in CRISPR technology, we cannot design the guide RNA accurately without a complete sequence of the genome. And that highlight why we should do genome sequencing project and why Kitake X is being used for study purpose as a research model is because it has a lot of advantages in research purpose, especially when Kitake X has a very short life cycle, which is around 9 weeks, compared to other varieties such as Nippon Bear. And based on phylogenetic approach, we can say that Kitake X and Nipo-Barre are very closely related to each other. And in a nutshell, for upcoming food crises that may happen in the future, scientists should intensify effort in rice research, as we know that rice is one of the staple food in the world. And before we doing any research, it is compulsory to do genome sequencing. That's all from us. Thank you.